With the prevalence of filters and beauty effects on social media platforms, do we really believe that the face under the filter is our true self? People like using filters because they're used to how they look after using them. Our brains are actually very sensitive to our own faces and can recognize ourselves after using filters. We reviewed relevant paper, which indicates that filters serve as a highly idealized tool for self-presentation. The more one retouches images, the more likely one becomes to view oneself through the lens of external perceptions. Frequent image filtering alters the way individuals perceive themselves.
Hence, our research question is labeled as follows. Q1. Do filtered and unfiltered self-faces have differences in EEG responses? Q2. Do people who frequently use filters show stronger EEG responses with their filtered faces? We first searched for previous studies on filters and on the neural processing of self-related appearance, trustworthiness, and attractiveness. Then we inspected our ERP waveforms, scalp topographies, and statistical results, finally defined the late ROI. In this study, we first proposed an E-prime experiment to examine how people respond to filtered and unfiltered selfies. Our design used selfies as the main stimuli. To control facial expression, we divided them into smiling and normal selfies. Each group included both filtered and unfiltered images. The task had 3 steps. First, participants saw one target selfie and kept this person in mind. Second, they viewed other selfies from the same group. Third, the target selfie appeared again, and participants judged whether the face looked real, trustworthy, and warm by pressing Y or N. The purpose was to compare responses to filtered and unfiltered selfies. However, for the final data collection, we used the standardized E-prime program provided by the instructor. Therefore, the EEG data and final analysis were based on the teacher's task, while our design served as the conceptual plan. We also collected questionnaire data from each participant. The questionnaire had 2 5-point Likert scale questions. The first question measured how often the participant used photo filters over the past 3 months. The second question measured the participant's satisfaction with their appearance. For filter use frequency, 1 meant almost never and 5 meant very often. For appearance satisfaction, 1 meant very unsatisfied and 5 meant very satisfied. In the final analysis, filter use frequency was the main questionnaire variable. We added this score to the EEG data and tested whether it was related to the EEG filter effect score. This allowed us to examine whether frequent filter users showed stronger neural responses to filtered self-images. The MATLAB code processed the EEG data in 6 main parts, but our analysis mainly used the output from Part 6. Parts 1 to 5 prepared the EEG data. The code separated different conditions, applied baseline correction, renamed event triggers, generated ERP/Taupoplot visualizations, and extracted ERP measures across participants, conditions, channels, and time points. Part 6 was the most important for our final analysis. It exported the single trial EEG data into an Excel file. The exported table included participant ID, gender, epoch number, picture number, trial type, self-condition, relevance condition, and the ROI value. The ROI value was the key EEG measure. It represented the average ERP amplitude for each trial in the selected ROI window. In this study, the ROI was defined as the mean ERP amplitude across CP1, CP2, and PZ within the 300 to 500 milliseconds time window. CP1, CP2, and PZ were selected because they cover the centroparietal area, which is commonly used to examine late positive ERP activity, such as P300/P3B-like responses. However, the grand averages sheet did not include full waveform data for CP1 and CP2. Therefore, we used PZ as the closest available electrode to visualize the ERP waveform. We selected the 300 to 500 milliseconds time window because it corresponds to a late positive ERP component, which is usually related to attention allocation and stimulus evaluation. Since our research question focused on whether participants processed the filtered self more strongly, this late positive time window was used as an index of neural processing strength. Due to the structure of the exported data, we did not have full-time series ERP waveforms for the filtered self and unfiltered self conditions. Therefore, The ERP waveform figure was based on the available grand average data for the relevance conditions at PZ. This figure was mainly used to show the general time course of the ERP response, while the actual statistical analysis was still based on the extracted ROI values for filtered self and unfiltered self. Processing step, we used the single trial sheet from data out. First, we only kept self-related trials because our research question focused on the filtered self. Then, we created a new variable called self-filter condition. The original variable self in trial had 4 labels. We recoded self-filter smiles and self-filter no smile as filtered self. We recoded self-no filter smiles and self-no filter no smile as unfiltered self. This reduced the data into 2 main conditions, filtered self and unfiltered self. This was necessary because our analysis focused on the effect of filtering, not on facial expression. So, after recoding, we could compare the EEG response between filtered and unfiltered self-images. After creating the self-filter condition variable, we calculated the average ROI for each participant. The original data were trial-level data, so each participant had many rows. However, our statistical analysis should be based on participants, not individual trials. Therefore, we used a pivot table to average the ROI values for each participant. We placed participant ID in the rows, self-filter condition in the columns, and ROI in the values area. The value was set as average of ROI, not sum. This gave us 2 values for each participant, 1 average ROI for filtered self, and 1 average ROI for unfiltered self. These 2 values were then used for the later paired t-test and correlation analysis. After averaging the ROI values, we created the final data table for Jamovi. In this table, each row represents 1 participant. We included 3 EEG variables, ROI underscore filtered self, ROI underscore unfiltered self, and ROI underscore filter effect. The filter effect was calculated as ROI underscore filtered self minus ROI underscore unfiltered self. This value shows how much stronger the EEG response was for filtered self-images compared with unfiltered self-images. Then, we added the questionnaire scores. Filter use frequency measured how often participants used filters on photos. Appearance satisfaction measured how satisfied they were with their own appearance. Finally, we imported this table into Jamovi to run the paired t-test and Spearman correlation. In this analysis, we compared the EEG response to filtered and unfiltered self-faces. We used a paired samples t-test because both conditions were measured from the same participants. The result was not significant, TT11 equals 0.017, P equals 0.986. The mean RMOI amplitudes were also very similar, 0.902 for filtered self-faces and 0.899 for unfiltered self-faces. So, filtered self-faces did not show a stronger EEG response than unfiltered self-faces at the group level. Here, we examined whether filter use frequency was associated with the EEG filter effect. The filter effect was calculated as ROI underscore filtered self minus ROI underscore unfiltered self. The Spearman correlation was positive, RO equals 0.273, which means frequent filter users tended to show a larger EEG response to filtered self-faces. However, the result was not statistically significant, P equals 0.391. So, this is only a weak trend. It does not provide strong evidence that frequent filter users have stronger EEG responses to filtered self-faces. The original question focused on the subjective experience, whether people who frequently use filters feel that their filtered self is more real, more approachable, and more trustworthy. However, the current questionnaire did not directly collect ratings of realness, approachability, or trustworthiness. Therefore, we could not directly test this question. As a result, we adjusted the analysis to a more conservative question, whether people who frequently use filters show stronger neural processing of their filtered self. Therefore, the present analysis should be interpreted as an indirect neural test of the original idea, rather than a direct test of perceived realness, approachability, or trustworthiness. This is the end of our presentation. Thank you for listening.